29 people have joined, so you can start now.
Okay, Bismillahirrahmanirrahim. Good morning. Maybe we just give you a few more minutes for people to join.
Khalid, there is an icon appearing on your screen, so can you move that one?
Okay. Yeah.
33 joined now.
Can you please log in? Yeah. Please log in as well. So I'll just start. Yeah, 35 people have joined. Let's get to 50, then we start.
Just give 1 or 2 more minutes, and then I think you can start.
I'll just quickly send these emails to these guys sometime. Maybe it's sitting in the inbox. I'm going to send the link again so that they can join straight away now. There's the link, maybe for easier option. Okay. Shall I start? Okay. Okay, good morning, everybody. My name is Khalid Mahmood. I'm just going to see if everything is working fine. Okay. Okay, how many people have 46? I'm just going to make a start. Farooq, good to go?
So just switch to the presentation mode and then just start now, please.
Okay. Okay. So good morning, everybody. Good afternoon in Pakistan. So my name is Khalid Mahmood. Today's session is organized by UPSIGN. You may already know what is UPSIGN. UPSIGN is a network of UK, Pakistan's scientists and innovators who would like to work together to address the issues of sustainable development in Pakistan. Sorry. Okay, so today's session we have organized on the Pakistan's challenge around food security and the issues and threats we face from pests and diseases due to climate change. So we have lined up very interesting speakers here. It would be me who would be chairing the session. We have got Professor Tariq Butt from University of Swansea, Professor John West from Rothamsted Research, Professor Naresh Magan from Cranfield University, Dr. Shahid Mansoor from NIBGE, Pakistan, and Dr. Belinda Luke from CABI. So before we start, just to give a bit of, I would say, the rules of today. We're trying to keep it very straightforward. We like to finish in under 1 hour. So first, and most of you have already turned off your microphones and cameras. So please keep them turned off during the session. So please look into your emails. We have sent out a survey form. I just sent it again today. So you need to fill out this form. And in this form, at the bottom, there is a question answer session. Question. So you can ask any particular question to a specific speaker. Now you can also use this chat box for asking the questions, but these questions will be asked, would be answered at that. So our plan is to have them from 10:50 Pakistan time, I would say 2:50. So we'll answer these questions. Speakers, they all know we have to stay within 10 minutes. And then at the end, please be respectful. So we should take this opportunity for learning. So there is nothing right or wrong. It's more like a research is always trying to find the refined knowledge to the next stage. So I'll just crack on. So why are we talking about Pakistan and Pakistan's food security? As you know, in Pakistan, agriculture plays an important role. So under 20% of contribution coming from agriculture in the GDP. So in terms of employment, its role is under 39%. And in terms of the rural engagement, so around 60% of the people in the rural areas, they are engaged in agriculture sector. And agriculture plays an important role in overall exports. So 58% of exports coming out of agriculture. And this sector is also dominated by women in terms of labor force, in terms of looking after the day-to-day affairs of agriculture. So 39%. So in Pakistan, this data is coming from last year. So how much hectare of every crop is grown in Pakistan. You can see from this chart, around 9 million hectares of wheat is grown in the country. Rice is grown on a 2.6 million hectares. Third most crop in terms of revenue, I would say, it's cotton. It's not food security crop, but it's a fiber crop, but it's playing an important role in terms of generating income for the country. And recently, the maize has taken over. So maize area is 1.7 million hectares. And after maize, we have got sugarcane, which is 1.5 million hectares. I don't know why it's not showing up here. So I just made some interesting comparison where Pakistan is in terms of its yield. So this chart shows the yield ton per hectare. So if you look at wheat, so the Pakistan is achieving around 2.7 million tons per hectare. Rice is 2.4 million ton per hectare. Cotton is 0.75, sugarcane 57 million ton per hectare. Maize is 4 million ton per hectare. But if you look at the world average, so more or less, I would say Pakistan is behind the world average. And if you want to look at in the middle, the countries which are the leading in terms of crop productivity, so New Zealand is in top in wheat. And we have, I would say, if you look at the UK, so UK is getting around 8.5 million tons per hectare. Then Australia in terms of rice, this rice is coarse rice. Australia is good in cotton as well. So it's interesting numbers to sort of set a scene. So we have many issues at the moment in Pakistan coming out of plant diseases, pests and diseases. The most important one at the moment is locust. This number I looked up is maybe slightly outdated. It says 21 districts. This morning I saw there are 33 districts. But the whole idea is to say we are getting issues from sort of pests which we haven't seen in the recent history. So locust is the one, very important. So in terms of the post-harvest losses, fruits and vegetables, they get the biggest burn. 53% of losses coming off fruits and veg. Cereals, 24%. Rice, 16%. And other food grains, 10%. So we're talking today sort of the issue of the post-harvest, and then it comes with the issues around mycotoxins and the use of pesticides to control these diseases. So it affects the human health, it affects the livestock health, and affects our ecosystem, our water and our air quality, and also affects our economy in terms of exports. So Pakistan is not able to export some of its products, especially rice recently. There was issue with one of the pesticide tricyclazole, which was banned, or the limits been in EU. So it's a huge food safety challenge. So we have to sort of keep an eye on how we can overcome. This is where our speakers will go in more details. I'm just giving an overview here. So I would say pesticides are a blessing, but at the same they are a curse as well. So how do we address this issue? This is where this whole seminar is today. Our webinar is all about. So I'll just bring it to our next speaker, Professor Tariq Butt, who's at University of Swansea. Professor Tariq Butt, would you please turn your mic on? So just to explain a bit about Professor Tariq Butt, he's the lead in UPSIGN as well, so in this UK-Pakistan Science and Innovation Network on the Agri Food side. And he has got 40 years of experience working on biopesticides, pathogenic fungi, and looking at chemicals and other interesting aspects of biocontrol. So today he's with us, and over to Professor Tariq Butt.
Okay, Khalid, thank you. So if we can go to the first slide. Okay, so this is just going to set the scene as to the pesticide use and some of the problems Pakistan faces regarding the use of chemicals. So first of all, there's no denying that pesticides play a major role in crop protection, and currently it's projected that Pakistan has a market reach for pesticides reaching about 350 million. This is by 2025, and it is without a doubt one of the major uses of pesticides in Asia, especially insecticides. In fact, the insecticides account for 58% of the sales. Next slide, please. Next. So the synthetic chemicals, the synthetic insecticides, this has captured 98% of the insecticide market, while biopesticides, and this is probably predominantly BT, they've captured only 2%. The problem in Pakistan is that many pesticides used there are banned or restricted in use in Europe, and probably 40%, more than 40% of pesticides counterfeit are produced locally or imported. In fact, the government of Pakistan has been taking measures for years now to try and curb the sale of fake or substandard pesticides. Next slide, please. If you look at the figures, the sales over time, what's very, very clear is that the pesticide use has increased exponentially. I couldn't get the latest figures of use, but you can see from this graph just over a few years how this has increased many, many fold. And the many drivers for this, one is because the area of cultivation is increasing, the crop area is increasing, but also because of the development of resistance in pest populations. Next slide, please. Now, the thing is, the pesticide use has its consequences. Next. So let's go through very quickly the drawbacks of chemical pesticides. It harms human health. We know it's linked with cancers, Parkinson's disease, allergies, and so on. It affects animal health, results in abortions, results in endocrine disorders. It kills pollinators, and this is quite important because the pollinators play a major role in improving yield. And according to the USDA figures, pollinators contribute to the global economy of something like 217 billion worldwide. Khalid had mentioned already that how the pesticide, can you go back please? They affect the exports, reduce the income and bad reputation. It harms the environment, reduces biodiversity, pollutes the environment, and very importantly, it results in resistance, which results in reduced yields and reduced income. Next, please. So focusing on insecticide resistance, next. And some of you are probably familiar, those in Pakistan are familiar, but not just Pakistan, and many other countries where you've got polyphagous pests, a number of polyphagous pests which have developed resistance to insecticides, such as Bemisia tobacci, the whitefly, Helicoverpa, Spodoptera elytra, Aphis gallipii. There's a whole host, the list is endless as to the number of pests that have developed resistance over time. Next, please. Now, the thing is that it's understandable, and it's a knee-jerk reaction of the grower, and the response is to increase the frequency of spraying. It can be from 2 to 13, more than 13 times. I know in some places probably a lot more than that. And they also increase the concentration, the dose of the chemical. So in other words, it's less diluted, and hoping that it's going to be more potent. Now, the net result is that the operator is exposed to these chemicals, so their health is at risk. You've got a lot of residues in the environment, particularly the crops, and you've got you're exacerbating the problem of resistance, built of resistance in pest populations, and there is obviously the damage to the environment. Next, please. And with this excessive use of chemicals, you can understand that there are going to be consequences, which impact on human health. And I've only listed a few examples here. People don't realize that in fact there's a lot more impacts. Cancer is one, Parkinson's disease, fetal development, respiratory disorders, skin allergies, and so on. And this poisoning is underreported or misdiagnosed. If you're not feeling well, a doctor may not put it down to some sort of pesticide poisoning, but to some other factor. But what we do know is that there are elevated biomarkers for these pesticides in farm workers and the general public. So we know that some of these chemicals are drifting through or somehow coming through the food chain and affecting the general public. And the thing is that you can also see that if you do analysis of residues, that more than 50% of the produce exceed the maximum residue levels, and with more than 20% of these exceeding the national and EU levels. So the chemicals are having a devastating effect on human health. And now some of the misuse of these pesticides, you could say, is partly due to poor education and training. A lot of the farmers cannot read or write Urdu, and obviously they're not fluent in English. So most of the safety data sheets are in English, and most of the farmers think the pesticides are safe. Next slide, please. So I'd like to finish off really summarizing what the major food challenge that Pakistan faces, that is how to achieve sustainable food production with reduced chemical pesticide inputs. Thank you.
Thank you very much, Professor Butt. Now we are, I think, just in time. So, John, are you ready now?
Yes, sorry, just unmuting.
Just do a quick introduction for John West. Professor John West is a senior research scientist at Rothamsted. You all may be knowing Rothamsted is one of the old agriculture research institute in the living planet, I would say. And I also worked at Rothamsted, so I didn't mention in the beginning. So I worked with Professor John West, and John West has a extensive experience in applied multidisciplinary crop protection, I would say, techniques, projects, I would say. He has got PhD in plant pathology from University of Reading, and he's now leading in at Rothamsted a smart crop protection program as well, so working with other colleagues. So he has got 2 patents, 8 book chapters, and over 50 papers, I would say. So over to John. Thank you so much, John, for joining today.
Thank you very much, Khalid, for the invitation, and apologies that I was unable to attend the workshop in Islamabad in March, which was due to the COVID-19 situation. But anyway, I'm glad I can attend this one. And this slide shows where Rothamsted is, just north of London in the UK. My purpose today is to explain the range of crop protection work that we do, which we hope we can develop as a collaborative project with people in Pakistan, academics and various commercial organisations as well. We work on a range of different crops, and those are arable crops and horticultural crops as well, and vegetables. In addition to my work on which focuses on diseases, colleagues at Rothamsted also work on a range of pests as well. You can see on the left-hand side, aphids are a problem in the UK for spreading viruses, but also it's possible to use the same equipment to detect larger insects like locusts. In the centre, some of these very high-tech tools are shown, things like insect suction traps, vertical-looking radar. It's possible to use existing weather radar to detect swarms of insects as well. And also automated spore traps are in development as well to make a detection of airborne spores. Now, a feature of all of these devices that are becoming smarter and smarter is that they can give information in near to real time. They give an indication of the geography where the problem is located. They quite often work in networks so that the information from each device is reinforced by the information coming from those devices around them. And then also we can add on a whole load of mathematical tools to do things like predict the trajectory of a swarm of locusts in terms of which direction it's going, and produce risk maps of where diseases or pests might be about to attack. Next slide, please, Khalid. So this focuses now on plant pathogens, and it actually shows a continuum of different monitoring approaches that we can use. And I've been involved in all of these different things, going from the bottom left, satellite imagery and drone inspection, which allows inspection of very large areas of land, but not always with the sufficiently detailed resolution to be able to pinpoint exactly what a problem might be. We can also do in-field walking to look for problems, and that can be backed up with lab or even in-field analysis using portable diagnostic tests to confirm the presence of a disease, either at very early stages of infection or even pre-symptomatic stages. We can also use weather-based forecasts, and the area I'm mostly going to talk about is inoculum-based forecasts. This is detection of the inoculum before it actually infects, and that's things like airborne spores, but it can also be inoculum present in soil or on crop debris that you can test for. And sorry, Khalid, yeah, thank you. If the key point is the top right-hand side of this graph gives us techniques that allow better preventative control of infection, because it's actually detecting the presence of the pathogen before it actually infects. And this, just following on from Tariq Butt's presentation, it's increasingly important if we're going to use biological control that we get the timing of our control methods absolutely right, and so better monitoring will help us to do that. Okay, next slide please. So for sampling for airborne spores, we have traditionally used a range of different devices. Some of these can operate for a whole week at a time, others operate for maybe just one day. And we can use different downstream methods to analyse the sample to see what pathogens were present. We used to use microscopy, and increasingly that is actually starting to become automated as well with image analysis methods. We can use immunological methods to do a dipstick or lateral flow device type test on the sample of spores that can be just made into a drop of liquid and put onto the device. But my work is mostly focused on DNA-based methods, and we can use new diagnostic tests like LAMP assays to do a very rapid on-site test for the presence of a pathogen in an air sample for that day. And we can also use portable sequencing devices like a MinION, which is quite expensive, but it allows us to detect hundreds of different pathogens, including animal diseases as well, that might be present in the air. And so that, although it's expensive at the moment, could be an extremely powerful tool for the future. Next slide, please, Khalid. So this shows a typical output. It's a little bit busy, but this is actually graphs showing the amounts of a pathogen called Sclerotinia as airborne spores trapped by the type of spore trap you can see on the top left side of the screen. That's a Burkhardt 7-day spore trap. And each week samples from those traps were sent to a lab and we analysed for the DNA of the Sclerotinia. And we could then find out what happened at the different sites. And you'll notice shown by an arrow is a big peak of spores at one of the sites, which was in Herefordshire. And we know that that peak of spores caused 20% of oilseed rape or canola plants to become infected. And the key thing is we didn't have that information in time to produce an application of a crop protection product because the sample had to be sent to a lab, and that caused a delay. And so the next slide actually shows, next slide please, that actually shows the new development to address that problem, which is near real-time automated analysis of what spores are in the air. So we now have a device that samples spores into the air, and at the end of each day it pipettes a sample of the mashed up spores into tubes with dried reagents present, and does a fluorescence assay, and the results are sent out by mobile phone text. And so this allows us to detect a range of different pathogens that we've already got assays for. You can see on the right hand side some of those. And we've also, in a current project, we're adding a lateral flow device capability as well. So inside the box we'll also be able to do a lateral flow test. That's an antibody based test for other pathogens as well. So we'll be able to do many different tests for different pathogens inside the box. Next slide, please. So in addition to the automated spore trap, there are still some quite simple devices that are cheap that we can use as well. And actually one thing that is promising for coverage of large geographies is to have a mobile spore trap. And what you can see on the left hand side is a device that's just attached to the roof of a car. And this actually has the air rammed into the top of the device and it's sucked then down into a lower chamber where the spores separate from the air and fall down, in this case, onto detached leaves of wheat plants, which we can then incubate to see which varieties are likely to become infected with diseases like powdery mildew or rust. And so that can also be used in conjunction with a diagnostic test. And on the right hand side is a similar device used by colleagues in Australia. And they've developed a way to get the air to deposit spores into small sample tubes on a carousel. And as the vehicle drives into different regions on a map, the carousel was switched to a different tube. And as they journey back into a previously visited region, the carousel will return to the correct tube for that region. So you can get a geographic analysis of which pathogens are present in different locations. Next slide. So lastly, in addition to sampling and doing a diagnostic test for things, I mentioned earlier from the satellite and drone imagery, it's possible to use optical sensing to detect all sorts of stresses. It can be drought stress, insect damage, and also diseases and nutrient deficiencies as well. A simple method to do that is to just look at the quality of light from the stressed plants compared to healthy plants, and there are ways of working out algorithms to identify particular stresses. In some cases, it's necessary to use an imaging method where a focused image is collected, and then image analysis methods can be used to identify the presence of disease and map where it is. And we've also been using in the past fluorescence measurements under UV light to detect very, very early stages of infection. And I'd be happy to collaborate with anyone on any of those kinds of technologies, the imaging and also the spore detection. So thank you very much for your attention, and thank you also to these collaborators and funders.
Thank you very much. Thank you, John, for your time and very exciting presentation. There's a lot more to learn, and just in these 10 minutes, we'll ask you some questions later on. So now I'm just going over to Professor Naresh Magan, who's a professor at University of Cranfield. Naresh is doing interesting work on post-harvest losses. Last 20 years, last 40 years, I would say. So looking at the issues around mycotoxins coming in the food chain. So over to Naresh. Can you see the screens, everybody? Farouk, can you see the screen?
No, not can't see this.
Moment.
Can't see in the screen, Khalid. No?
No.
Okay, let me then. Okay, which screen I need to.
Do you want me to share the screen?
Yes, please, if you can. Yeah.
I'm not sure we can hear Naresh either. I don't know if you need to enable his microphone.
Can we look at Naresh? Naresh.
So can everyone see the screen now?
Yeah, I can see the screen. Thank you.
Okay.
We need to make the Naresh a presenter. Let me make him a presenter. Naresh, you are a presenter now.
Can you hear me?
Yes,
we can hear you.
Go ahead. Okay.
Share video. Okay.
So now we can see the screen as well here.
Okay. I don't know what's happened with my video. Can you hear me?
Yeah, we can hear you. Yes, we can see the screen here as well. So we're on the first page.
No.
So if you want to.
Naresh, you can start with audio because we got the screen and we can keep on moving the slides.
Maybe we're having some issues here. Naresh? Okay, Naresh, I'm gonna make a presenter again. Naresh, can you hear us? Oh, sorry. No. Sorry. Okay. Just give me a second. Naresh, can you hear us? Maybe it's gone. Farouk, can you go to the next slide at least so that, please. He's joining now, Naresh.
Do you want me to go to Naresh slide or shall we go back to main presentation?
So I was just thinking if there's an issue with Naresh, then we can just skip through here. Just give him a few seconds here. Can you see him in the window? Sorry guys for this technology.
Khalid, just give a call to Naresh and then explain if anybody can phone in, and then we can just
So
continue with
shall
that
we get
presentation.
the next speaker then? So let's get Belinda then. Belinda Clark. Okay, shall I share Belinda?
Yeah, share. Yes, please.
Okay, I will do. Okay. Okay, share my screen, Belinda. Okay. So just would like to introduce here Dr. Belinda Luke. She's working with CABI as a senior insect pathologist, and she has over 23 years of experience working in biopesticides from isolation all the way to registration. Can you see my screens, Belinda, here?
Yes, I can.
Okay. And she has particular interest in mass production, formulation, and quality control. Belinda was in Pakistan earlier in March on the global development workshops. So you all know CABI has a presence in Pakistan. So Belinda has a lot more to offer to this group and for Pakistan as well. So over to you, Belinda. Please.
Thank you. Well, hello, everybody. Next slide, please. So today I'm just going to be talking about biopesticides. And first of all, I'm going to explain what biopesticides are because they cover a whole range of different organisms. Then I'm going to specifically look at microbial pesticides. I'm just going to see how they're useful. And then I'm just going to give 3 different examples of biopesticides, and then I'll just summarize it at the end. Next slide, please. Thank you. So in the UK, biopesticides are described as plant protection products which contain biological control agents. These agents can be microbials. For example, the top picture is an insect infected with fungus. Pheromones, which is the middle picture, and normally mating disruptors, things like that. And then plant extracts, which is the bottom picture, which is neem oil is the most widely known for that. And they can be used in agriculture, horticulture, or in the home garden pesticides. We're currently seeing that the biopesticide market is expanding because you've got resistance, chemicals being banned, and a desire from the end consumer that they want a healthier food. So within biopesticides, you have something called microbial pesticides, which are broken down into 4 further categories. You've got bacteria, Bt being the most common one, which is good for controlling caterpillars, flies, and mosquitoes. You've got fungi, which kills locusts along with other insects, which is very apt for Pakistan at the moment. But you also have fungi like trichoderma that can kill plant diseases. Nematodes, which is the picture in the middle at the bottom, very good for soil-dwelling insects and for slug control. And you have viruses such as Helicobacter, which is also good for caterpillar control. Next slide, please. So enteropathogenic fungi, this is just looking at fungi at the moment, has lots of advantages. It's got shorter pre-harvest and re-entry intervals. It's less toxic to wildlife, natural enemies, and humans. To date, there's been no record of resistance to mycoinsecticides, and it can be used in organic farming, which means that in organic farming, you get a higher price for your end product. Next slide, please. So something that CABI, along with other partners, worked on, well, it started in 1989, is green mussel, which is a fungus that controls locusts and grasshoppers. So obviously this is really important at the moment in Pakistan because you are being devastated by this crop. Now, this commercial product, we worked from finding the isolate, this is based in Africa, right through to working with commercial companies to help register it. The product is called green mussel. It's environmentally friendly and it's very effective at controlling locusts and grasshoppers. And it's now being produced and sold by Elephant there out of Morocco. Next slide, please. And I just want to give you briefly how green mussel works because it's very different to a chemical pesticide. You apply it exactly the same as a chemical pesticide. You spray it, whether that's aerial or tractor-based sprayers, whatever it may be. The spores come into contact with the locust. They can either do that directly or they can pick it up from vegetation that's been sprayed. Once the spore's on the locust, it then starts to penetrate the locust, which it does through physical pressure, just forcing its way inside. It then grows within the locust, and then eventually the locust will die because it's being eaten from the inside out. The locust, dead locust, will tend to turn a pinky colour, and if environmental conditions, can you just click next please, are favourable, then you will get external sporulation. Oh, yeah, thank you. Which then means spores are released that can then go on and infect other locusts. And that's what we call secondary recycling. But you will notice here in the middle, it says on average 7 to 20 days to work in the field. And that is very much dependent on environmental parameters. So climate change will have a big effect on how biopesticides work. Next slide, please. And this is just looking at one project that we had in China, which was looking at control of the Chinese migratory locusts. And we've produced an app called BioSuccess, or in the process of developing it. If you look at the diagram on the left-hand side of the screen, you'll see that you have got a colour map put over China. At the bottom, it's pink, going through red, orange, yellows, greens, blues, and then at the top, black. So if you see the purple area at the bottom, if you then look to the bar on the right-hand side, you will see that it takes 14 days for 90% of the locust to die from the biopesticide that's been sprayed. But if you go higher up in China, where it's cooler temperatures, in the black area, it takes 40 days to kill 90% of the population. So depending on where you are, will depend on how quickly your bioproduct works. And not only that, that date was on the 15th of April. If you look at the picture on the right-hand side of the screen, on the 15th of September, you'll see that the colours have changed. And in fact, you can have quicker control in a lot more of the areas. So a lot more areas are pinks and purples. So half of that area now, you'll get 90% kill within 14 days. Next slide, please. Now switching to Bt transgenic cotton. So Bt is Bacillus thuringiensis. It's a bacteria that produces a natural toxin. What you can do is you can put this toxin within your cotton plant, and then you can sell your seeds. So when the cotton plant grows, the seeds contain the Bt toxin, which is very good at controlling helicobacter and other caterpillars. Now, Pakistan, cotton, in Pakistan is the most important cash crop, and accounting for 10% of the national GDP, and it's about 55% value of exports. So in theory, if you put Bt cotton on, it should result in less pesticide application, because effectively the pesticide is within the plant. There's been a very good paper that was produced in 2019 by Kusa et al. And that looked at farmers' health in relation to the use of Bt cotton. But I just want to draw out a few biopesticide-related issues from this. And that the first one that I noticed was that there's an unregulated seed market. Bt cotton entered Pakistan before officially it was allowed to. And in the paper, it noted that 17% of farmers who thought they had bought Bt cotton, in fact, just bought normal cotton seeds. So their crops were not protected as they thought they were. This can really damage the reputation of biocontrol products. On the converse side, 55% of the farmers, this is just in Punjab, thought they had bought normal cotton. In fact, they had Bt cotton. Well, you think, oh, well, that's quite good. Not really, because those farmers are still putting on chemical pesticides, perhaps unnecessarily. And the paper reported that for the true Bt adopters, on average, it saved about 33% of health costs. And that's not just the cost of people being ill or not. It looked in transport costs to go to hospitals, cost of medicine, cost of not being able to work because you were sick in bed. And if you extrapolate that up to the whole of Pakistan, you could have annually a saving of $6.9 million US in reduced health costs. Next slide, please. So this is a project that CABI have been working on with Pakistan partners and US partners, and it's about controlling aflatoxins in maize. So aflatoxin is the toxin produced by a fungus, and it gets passed on in the food chain and is harmful to humans and to animals. So what this project did is it found a native non-toxin producing fungus, and you release that into the environment. It outcompetes the toxin producing fungus, so therefore you're not getting that on your crops. As far as we're aware, this is the first public-private partnership leading to registration of a biocontrol product. There were 3 different compartments of the project. One is obviously assessing the effectiveness of aflatoxin, but perhaps more importantly is we were looking at the training of the national scientists and the maize growers, because it works differently to a chemical. We need to explain the process so that they're aware and they know what to look for. And then also the registration of aflatoxin is the first of its kind in Pakistan. And registration is a really important issue. Registration for biopesticides in Pakistan is new, it's in its infancy, and I see this as a great opportunity for Pakistan. In the UK, when we try to register a biopesticide, it has to go through an adapted chemical system, and it does not work. It doesn't really work because the tests are very different for what you need, and it's very expensive. Pakistan have a great opportunity to develop a Biocontrol registration system, and I think if they could do that, it would be amazing. Next slide, please. Now, the impacts of that project, obviously you've got the agricultural impact, the farmers and the stakeholders, how to increase awareness. Trade, you can do more trade and more exports potentially because your maize is of better quality, and obviously the health impacts because those people aren't eating toxins and becoming ill. Next slide, please. So in summary, I would just like to say that Biocontrol is becoming available in Pakistan, and it will be a booming market within the next 5 to 10 years. Market regulations are needed to make sure that those Biocontrol products in the market are genuine and they're not being sold duds. Great opportunity in Pakistan to work on the registration process. Use experience of the European system, which doesn't work very well, to design one that could work really, really well. And also, as it's already been mentioned, CABI's already got a site in Pakistan, but I think there's great opportunities for Pakistan and UK to further collaborate, to use the extensive knowledge in Pakistan with biopesticide knowledge in UK to get more products on the market. Thank you.
Okay. Thank you so much, Dr. Belinda, for your inspirational talk. There's a lot more to learn, and they're looking at the scope for Pakistan in terms of increasing exports. It's fantastic. So thank you very much again. We will try again, Professor Naresh Magan. So Farooq, over to you.
I've shared the slide now. So
Yeah. Okay. Is Naresh ready? Naresh? So maybe we need to make Naresh presenter again. Just a second. Sorry guys, because we didn't use this system before. So we had actually a last minute change, because originally we had Zoom, but Zoom doesn't allow us to have more than 100 people. So, but okay, Naresh is here. I'm gonna make him a presenter. Naresh, you are a presenter now. Naresh?
Can you hear me?
Yes, we can hear you now. Yes, perfect.
Can you hear me?
Yes, we can hear you. So we are on the first page of your slide. Yes, go ahead. Yes, Naresh?
Can you hear me?
Yeah, we can hear you.
Okay, let's go to the next slide.
Farooq, next slide, please. Or can you make it bigger? Maybe then we will be able to see the entire slide. Thank you.
This summarizes some of the important mycotoxins which are present in different important commodities, including cereals and so on. Can you go to the next slide, please? It's important to remember that different grain commodities have different nutritional value. And the important thing is that when you have mycotoxigenic spoilage, you have nutritional losses. So this creates what I would call a double problem. Quality, and therefore the nutritional quality of your commodity changes, as well as you get significant impacts due to mycotoxin contamination. Of course, this we consider may increase under future climate change scenarios. So it's important to consider both aspects. Next slide, please. And just to show, I thought I would focus some work we've done. Rice, sorry. We've looked at analyzed rice, especially both paddy and brown rice, to compare the two. And we've isolated the fungi and we've quantified the different mycotoxigenic fungi present. And we found Fusarium species and Aspergillus flavus, which produces aflatoxins, and the Fusarium, which produces fumonisins. So let's go to the next two slides. Next slide, please. Thank you. This just shows you that if you have paddy rice or you have brown rice, if on your excess you've got protection
Can others hear? I think I can't hear. Maybe some.
I can hear.
You can hear. Okay, perfect.
Yeah, but I think we've lost Naresh. I can't hear Naresh Magan, unfortunately.
So let me try again, Naresh. Naresh. But Naresh is here. Naresh. Make you speaker, make you presenter. Naresh, can you hear
Okay,
us?
now, sorry. Can you go back to the last slide? Sorry. Okay, I was just going to say that we have similar data for fumonisins, and it again shows very low quality loss due to mycotoxigenic fungi. On mycotoxin contamination. Next slide, please.
Naresh, I think your voice been breaking up. So maybe the issues with the signal. So we will share the slides with you. I mean, if people have a bit of difficulty in hearing today, so we'll share the slides. Sorry for the voice issue here. I think Naresh is just going to go through these slides quickly. So, Farouk, I think Naresh has an issue with the connection.
I'm just slowly going through the slides so people can see the slides and.
Okay. Okay. So maybe you could sort of I don't know, maybe someone could step in.
I think it's a bit technical study, so I think.
Okay. Okay. Maybe just so I think we'll share these slides. I will just maybe stop. If just finish these slides, scroll through, and then we'll
Khalid,
start
it's John West here. I think we've unfortunately got an internet issue that meant we've lost Naresh's sound. I think what he wanted to show was some data from CO2 monitoring, which is an indicator of the presence of spoilage organisms in the grain store. And I think this slide is showing some other work that he's developed. Obviously, I think this is something that individuals could contact Naresh on in order to get better up-to-date information. And I think it shows some really nice techniques measuring things like relative humidity and also understanding the impact of how to store certain commodities in a way that doesn't cause the right conditions for spoilage organisms to grow. So I don't want to say too much, you know, on Naresh's slides, but he's the expert to contact. Okay.
Next. Okay, so I think we can now go over to sort of yeah. I think there are a couple of slides more. Farouk, just scroll them through, and then I think we can just open up some questions. I'm just conscious that we're just getting to an hour now. So yeah, excellent. So thank you very much. So we have got some burning questions. One question is asked for John West. So can we use this alert system for rust diseases for wheat? This was a question asked by a gentleman, I think, wheat rust from Imtiaz, Pakistan. So John, could you please answer this question? John West.
Yeah, sorry, can you just repeat the question?
So they asked, any thoughts on the early warning system for rust in wheat?
Yes, well, I noticed Belinda replied also because she or CABI have a project doing something similar in China, and there are a range of different methods that can be used. So at Rothamsted, we've been developing the automated spore trap, which makes a detection of the spores that can blow very long distances in the wind. And so it's possible to make quite an early detection very early in the season when the wind blowing from a certain direction might be introducing spores to a region, and that means that you can get an indication of imminent disease in time for an application of a crop protection product, whether that's a fungicide or a biological control agent. And there are also some methods using satellite imagery to observe where rust is currently present. And then if you tack onto that some mathematical techniques combined with an understanding of the wind direction, it's possible to do what's called forward tracking to work out where the spores might be blowing to next. So there are a range of different approaches. The one I would advocate as a really good tried and tested method is the spore detection using which we can use all sorts of spore traps for, some relatively cheap that don't have to be automated, but can be just used with a manual test done at the end of each day. Okay.
Thank you very much, John. So there was another question asked by Shahid. It's around the collar rot disease in citrus. So any thoughts on this, John West?
Yeah, I don't actually. I've never worked on collar rot in citrus. I don't know whether this is a basidiomycete like our malaria, or whether it's a different pathogen. So maybe if they can contact me by email, I'll be able to do a bit of research and then make some recommendations. Certainly, if it is what I think it is, which is a basidiomycete, I think it spreads through the soil by the mycelium growing in strands called rhizomorphs. So it's a question of removing any affected trees, so you actually completely remove them with as much root material as possible, and leave that area fallow for a while before replanting with a more resistant plant. There are ways you can add spent mushroom compost, which contains a whole load of antagonistic organisms in the compost, and you can dig that into the soil, and that can help to reduce that kind of pathogen. But again, I'm not entirely sure that I've got the correct pathogen that the question was about. So if they can contact me, I'll try to give us some information by email. Okay.
Thank you, John. There's a lot of questions around locust, and they are towards the direction of Belinda. So Belinda, people are asking about how biopesticides are effective in desert locust control. And yeah, please, over to you.
Okay, so the fungus that we found, Metarhizium, is very effective in killing only locusts and grasshoppers. So if it lands on a different insect, it won't infect it or kill it. So, for example, bees and pollinators are safe. And also, if animals eat infected locusts, it doesn't go up through the food chain and cause death of birds of prey who are ultimately at the top, or effects on humans if humans eat them. So they are safe in that respect. They're very good. If you formulate them right and you apply in the right way, so in Africa, they're applied in an oil formulation, ultra low volume, you can get very good control under those environmental conditions. And I appreciate that it's very hot in Pakistan, so it all comes down to timing. Make sure you put your pesticide on first thing in the morning. Don't do it in the hottest part of the day, for example. So we do know it can work. We do know that it really doesn't have that many side effects. I guess the biggest health issue would have to be concerned is the actual people who are applying it. So when you open up the bag of spores, it needs to be mixed. So it's a dusty consistency. So as with any dust, you need some sort of dust mask to get rid of that dust. Don't breathe it in. However, saying that, no one's been recorded of dying of Metarhizium. You may have a headache, you may have a bit of difficulty breathing temporarily, but there are no long-term health effects that have been documented so far with that.
Thank you very much. I'm just going to take last question, and then we'll wrap up the session for today. There's a question around aflatoxins have negative impact on human health. So I think Naresh is in a position to answer, but if Professor Tariq, you could answer a bit on this question.
Well, the thing is that mycotoxins in general have negative effects. I mean, aflatoxins can cause cancer. I mean, we in the West have had a screening program where you actually try to look for the toxins and make sure that they're not in the food chain. And Belinda, I think, mentioned already that there's been a massive program and a hugely successful program both in the US, and she's mentioned one which has been tested in Pakistan, where you're using a toxigenic-free strain of Aspergillus, to the whole purpose being to displace the toxigenic ones to stop the actual toxins entering the food chain. So the thing is that there's no doubt they cause cancer and a whole range of other problems, health problems. So I don't know if that's answered the question, but to give you a flavour of that this is not exactly a nice compound.
Thank you. There was another question around biocontrol in cotton, whitefly, the Mysia tabaci. Anyone who's the best? Is Belinda, back to you.
I don't really know much about BT because that's not my area of expertise. I do know that there has been reported, oh gosh, what am I trying to say? Resistance against BT. I'm afraid I don't know what the levels are within Pakistan.
Okay.
And it's in some ways it might be hard to judge because of this unregulated seed market. So the farmers may be told they're putting on such and such, but whether it is actually that, we don't know.
Okay. We have got here Dr. Shahid Mansoor from NIBGE. Maybe Shahid, you could answer this question. Shahid? Maybe overall control in whitefly in cotton. Shahid? Shahid Mansoor? Can you hear us? Maybe I need to make Shahid the presenter first. Just a second. Shahid.
Can I come in and say something here, Khalid?
Yes, please.
One of the things that I've been working with whitefly for many years, and I used to come to Pakistan, and a lot of the people are very young and may not remember this, but Pakistan used to lose something like $4 billion worth of cotton because of cotton leaf curl virus transmitted by whitefly. And even now, I think still they probably are losing roughly about $1 billion. And the main problem is resistance. It's quite extensive. It's a polyphagous pest. You find it on a wide range of crops. So you can imagine with the extensive use of chemicals, you're using mostly on cotton. Don't forget, 69% of the pesticides used in Pakistan are used on cotton. And so it's a heavy use of pesticides. But then the same pest can actually move on to okra, onto other commodities, and also some of the household plants. You have things like hibiscus, closely related as a malvaceae, same family as cotton and okra. So the thing is that the pest can survive and reinfest crops. So I think that there's a need for what you call a radical way of tackling this problem, because the same problems persisted now for more than 20 years, and it's not going away. I know some new chemicals have come on the market by Bayer, ketoenol-based compounds, but then if you look at the history of some of these chemicals, their resistance you get build up very quickly within 5 years. So that means we do need to rethink about how we go about developing integrated pest management programs for whitefly and the other pests which actually are resistant to insecticides as well.
Okay. Thank you. Very helpful, Tariq. We have got here Shahid Mansoor. Shahid, you want to chip in? Shahid? I'm just going to make Shahid again a presenter. Okay. Shahid Mansoor, can you hear us? No. So I would say today's discussion was more about sort of the issues Pakistan is facing with pests and diseases. And as our presenters, our speakers have given an overview about the challenge. The one thing which has come very clearly, become very clearly outstanding here, because we need to focus on surveillance systems, how we can develop a monitoring and surveillance system that can allow beforehand preparations, and also knowing what sort of armory is available. So it's not only the pesticides, the chemical pesticides, they're looking at alternatives, especially biopesticides, and making use of IPM. So these are the key strategies that we need to look in together. So I would say there would be some other series of seminars later in the month or next month or in coming months, which has been organized by UPSIGN. So I'm just going to show you quickly which are on in the pipeline, just to let you know, so that you can prepare yourself. There will be one specifically on locust control. So we haven't, because this is topic at the moment been covered by a lot of other organizations. We thought just to give a bit of time for others to look into it. But there would be one specifically on biopesticides, which Dr. Farooq Shah will be sending information fairly soon. So just quickly share my screen and let you know which other seminars are in the pipeline. So just a second. So we are looking at these topics. Locust control strategies would be the next one. Biopesticides specifically, going in more detail. And then we plan to do one on resilient breeding as well. And then would be a nutrition and health. As you know, with the COVID, this is a fantastic time for us to learn and share and engage with the partners in the UK. So there would be one specifically on remote sensing. I know some of our colleagues working in LUMS and in University of Agriculture, Faisalabad, so they would be very interested in this kind of discussion. There would be one seminar on animal health. So we had colleagues from University of Edinburgh and Roslin Institute who are happy to lead on that one, on what food waste, water resilience, vector monitoring, and control of zoonotic diseases. And then at the end, which is very topical these days, how blended learning could help to address the issue of education, but also knowledge exchange and helping the industry, and also in terms of improving the policy framework. So with that, I would say thank you very much again for attending this webinar. Sorry about the technology. We had a bit of issues this morning. So I hope next time we'll be able to overcome this challenge. So with this, say we would like to close this workshop with this webinar. Anything anybody from our speakers want to say before I close?
Yeah, John West here. So Khalid, thank you very much for organising it. Naresh, Magan did email me to apologise. I think he's had some internet problems today, but I hope anyone will be able to contact him by email with any specific questions. And one last brief thing, I quickly checked the colour rot of citrus. I got that wrong. This is the problem with common names. It's not armillaria, it's a species of Phytophthora. And so there are various chemical controls that similar to the ones that would control potato blight that would probably be effective. But I think the general advice I said about actually quarantining the location and avoiding moving plants around or spreading soil about within the citrus orchard will definitely help prevent it spreading. And there are also biological control options. I mentioned the spent mushroom compost. I think that would also have some activity as well against Phytophthora. So anyway, I hope that helps.
Yes, thank you very much, Khalid, for hosting this, and it was a very enjoyable webinar. Thank you very much indeed.
I'd just like to add that, and thank you for the interesting questions as well from our participants. Thank you.
Thank you. Thank you very much. Thank you very much, Farooq, for your help. Farooq is a co-lead on agri-food within UPSIGN. So those who have already known about UPSIGN, UPSIGN is the UK's Science Innovation Network. The whole idea is to bring the research on both countries working together to address the common issues in Pakistan. So the main framework that we are working on is UN Sustainable Goals. Agri-food, food security comes on its priority. So once again, thank you so much. Have a lovely, nice evening in Pakistan. Keep yourself safe. I know COVID is still an ongoing disease challenge in Pakistan at the moment, so please keep yourself safe. So thank you so much, and big clapping for the speakers. Thank you. Bye-bye.